Assalamu alaikum students, hoping you all will be fine. Today we will discuss about the lipids. Our major concern is the lipids. I'm following Leninger principles of biochemistry. It's 10th chapter in the 6th edition of this book. In this chapter, we will study the biological role of the lipids. We will study what are the structure of storage lipids, what are the structure of the membranous lipids, and what are the structure of the signaling lipids. So on the basis of their function, we are dividing the lipids into the three parts: storage lipids, membranous or structural lipids, and the signaling lipids. Lipids are actually the structurally diverse classes. You people know these are the compounds which are insoluble in water and soluble in the organic solvents. Their physical properties reflect that these are the hydrophobic in nature. On the basis of classification, they are divided into almost 7 classes. These are the organic molecules that are characterized by the low solubility in water. That is relatively relatively these are the hydrophobic compounds. Lipids are the major component. Lipids major components are the fatty acids. Uh, they are monocarboxylic acids. They may be straight chains, even number of carbon atoms, and they may contain even number of carbon atoms, or they may contain the odd number of carbon atoms. Fatty acids in the animal, fatty acids in the animal, or we can say the origin are those fatty acids which are originating from the animal. They are simple in the structure. They are straight chain, and they may contain 16 to 22 carbon or 0 to 6 double bonds in their carbon chain. Bacterial fatty acids, these are saturated compounds, and monoenoic acids, these are branched or contain cyclopropane ring. Plant fatty acids, these are more varied, have acetylenic bonds, epoxy bond, hydroxy or keto groups, or cyclopropane or cyclopentene ring. Or they may contain cyclopropene or cyclopentene rings. Biological role, they can perform several biological roles. They can perform the function of storage of energy. These compounds are actually the reduced compounds, and they have a lot of available energy. They are hydrophobic in nature, and may provide the good packing material. May provide the good packing material, like they may form the lipid bilayer of the cell. Insulation from the environment, low thermal conduct. They have the low thermal conductivity. In high heat capacity, they can absorb the heat. Mechanical protection, they can provide mechanical protection against the shocks, against or they can absorb the shocks. They are water repellent because they are hydrophobic in nature, so they are water repellent. Um in some diving birds, they provide prevention against excessive wetting of the bird's wings, excessive wetting to the bird wings, prevent loss of water via evaporations from the plants, buoyancy control, and acoustic in marine mammals. Marine mammal can float in the sea because of their high fat contents, because of their high lipid contents. Increased density while diving deep helps sinking. It may be just a hypothesis. Um, More functions, they provide structure to the membrane. Main structure of the cell membrane is composed on the lipid bilayer, cofactor of the enzymes. These are vitamin K. Example of this is the vitamin K, blood clotting formation, coenzyme Q, ATP, which is involved in the ATP synthesis in the mitochondria. These are the signaling molecule, like the paracrine hormone, which can act locally, which can act on their neighboring cells. Steroid hormones act body-wide. They Steroid hormones are made up of the lipids. Growth factors are made up of the lipids, and vitamin A and D are made up of the lipids. Pigments, these are present in the pigments. Color of the tomato, carrot, pumpkins, or in some in the some birds, we can see all these. And these may be they may perform the function of the antioxidants. Like for example, they are the components of the vitamin E. Here you can see the structure of canthoxanthine, which give the bright red color to the birds, and zeaxanthin, which can give the bright yellow color to the bird. So these color are because of the lipids. Classification of the lipids, we can, I already told you that we can classify the lipids on the basis of their functions. Majorly, we divided these lipids into the two forms. Number one is the storage lipids. Number two are the membranous lipids. In the storage lipids, the common storage lipids are the triglycerides, which are made up of the glycerol and the fatty acids. So it means glyceride mean that a glycerol molecule is attached with the three fatty acid groups. Then the membranous lipids. In the membranous lipids, there are three major forms: phospholipids, glycolipids, and the archaeal lipids. Phospholipids, glycolipids, and the archaeal lipids. The phospholipids is again divided into the two forms: glycerophospholipids and the sphingolipids. In the glycerophospholipid, phosphate and alcoholic group is attached, and in the sphingolipid, instead of the fatty acid, phosphate group and choline group is attached. And on the other hand, the third, in place of glycerol, there is the molecule sphingosine, while in the glycerophospholipid, the glycerol molecule is attached. Membrane lipid is second class of this type is the glycolipid, which can again be divided into the sphingolipids and the galactolipids. In the sphingolipid, the major molecule is the sphingosine, and here on the third position, mono or oligosaccharides are attached. In the galactolipids, there are two fatty acids attached with the glycerol, and on the third position, mono or disaccharides are attached. And the third one are the archaeal or archaeobacterial ether lipids. In this group, glycerol is attached with some other compound through the you can say the ether linkage. And mostly lipids are attached with the other compounds through the ester linkage, but in these lipids, the glycerol molecule is attached through the ether linkage. Fatty acids, these are the monocarboxylic acid. Carboxylic acid with the hydrocarbon chain contains between the 4 to 36 carbons. Almost all the natural fatty acids have an even number of carbons. Naturally occurring almost even number of carbons. Most natural fatty acids are the unbranched. They are further divided into the on the basis of the presence of double bonds in them. They may be saturated fatty acids, they may be monosaturated fatty acids, or they may be polyunsaturated fatty acids. Saturated, there is no double bond. Monounsaturated, one double bond is present between the carbon in the alkyl chains. And polyunsaturated, more than two. More than one double bonds in the alkyl chain. Animal fatty acid, I already told you that these fatty acids which are in any animal in origin, fatty acids from the animal origins. They contains the simple structure straight chain and 16 to 22 carbons. 0 to 6 double bonds are present. Bacterial fatty acids. These are saturated monoenoic acid, branched or contain cyclopropane lingering. Plants fatty acids more varied. They have more varied and having acetylenic bonds. They may have epoxy, hydroxy, or keto group. Cyclopropene or cyclopentene ring properties of the fatty acids based on the carboxylic and other functional group, uh, and their properties depend on carboxylic and the other functional group as well as on the degree of their saturation. Ester bond susceptibility to the hydrolysis. Ester bond can be hydrolyzed by treating with the acid. and this reaction may be reversible and can gain equilibrium. When we treat the fatty acid with the bases, this reaction is irreversible because in excess of base presence, acid exists as fully dissociated anions, having no tendency to react with the alcohol. So if we treat here, you can see if we treat the triglyceride with the sodium hydroxide. So because of this treatment, these because of this treatment, the glycerol will get the 3-hydroxyl group from the NaOH, and the alkyl chain here where the alkyl chain was, it will be attached with the sodium molecule. So when the sodium molecule will attach with these alkyl chain, they will make the three molecules of the soap. Fatty acid nomenclature, chain length, and fatty acids uh, we can identify by the symbols. We can identify the fatty acid by the symbols. This symbol tells us what is the number of carbon atom in in this fatty acid, what are the double bonds, and where the position of the double bond is. Chain length and the number of the double bonds separated by the double dot. This one. 16 carbon saturated palmitic acid abbreviated as 16 because this is saturated and contains no double bond, so it will be denoted as 16 is to 0. Oleic acid with the 18 carbon and one double bond will be represented as 18 is to 1. Position now the next is the position of the double bond. Position of the double bond specified to the carboxylic terminal. Specified to the remember this is very important. Specified to the carboxylic terminal. There may be two terminals, one carboxylic terminal and second one is the methyl terminal. Regarding the methyl terminal, I will explain you later, but right now for the general nomenclature of the fatty acids, we use the carboxylic terminal. Say for example, 20 carbon fatty acid with one double bond between the carbon number 9 and carbon number 10 and one double bond between the carbon number 12 and carbon number 13. It will be represented as 20 is to 2, and the sign delta will show, the sign delta will represent the position of double bond. Upon the delta, we will write 9 is to 12, 9, 12. So 9 and 12 position is the position where double bonds are present. This is the table regarding the fatty acid nomenclature. I advise you all to look on this table and to explain by yourself. Fatty acid nomenclature. Again, polyunsaturated fatty acid in which double bonds separated by the methylene group. Now I'm on the methylene group. Polyunsaturated fatty acid in which the methylene group is present. These are these can also be denoted as PUFAs, polyunsaturated fatty acids. So if we look from the methylene side, then most of the double bonds are present between the carbon 3 and carbon 4. Most of the double bonds are present between the carbon 3 and carbon 4. So if we look from the methylene group, and the double bond is present at the carbon number 3 and carbon number 4. Uh, such kind of uh, such kind of double bond are very important with respect to the nutrition point of view. Physiological role physiological role of the PUF is more related to the double bond near the methylene group. If we need to look the physiological role of the PFUs, PUFAs, this is very important, and we are interested to look on the nutritional point of view. This is very important to look on the methylene group. This carbon is called the methylene carbon is called the omega carbon and give number one. This carbon is called the omega carbon. And now, if we are looking for the methylene group, so this will be carbon number one. Fatty acids with the double bond between the carbon 3 and carbon 4 from methylene and are called the omega 3. If the double bond after the methylene group is on the third position, it will be omega 3 carbon. It will be omega 3 fatty acids. Fatty acid with the double bond between carbon 6 and carbon 7 from the methylene end are called the omega 6 fatty acids. Omega 3 fatty acids are essential nutrient. Humans need them but cannot synthesize them, including the ALA, DHAs, and EPAs. Um, Although DHA and EPA can be synthesized from the ALAs, but these are very important for the human. Here in this diagram, you can see here is the 18 is to 1 and the position number at the position number 9. If you can see here, the position number at the position number 9, octadenoic acid. Here the double bond is present, but this is the general classification, and this is the eicosapentaenoic acid (EPA). EPA. It contains, according to the according to the general classification, if you look from the carboxylic group, it is on the position number 5th. The first double bond is present on the position number 5th, second is on 8th, third one is on the 11, and the fourth one is on the 14, and the fifth one is on the 17 carbon. But if we look from the omega end, omega carbon is the carbon number 1, carbon number 2, and carbon number 3. This double bond is present between the carbon number 3 and carbon number 4. So this 20 is to 5 fatty lipid will be the omega 3 fatty acid. Solubility decreases as the chain length increases. You people know that these molecules are insoluble in water and soluble in the organic compounds. Their melting point also depend upon the chain length. If the chain length the melting point will decrease, and decreases. Melting point also decreases as the number of the double bond increases. The saturated chain tends to adopt extended conformation. The double bond in the natural unsaturated fatty acids are commonly in cis configuration. Uh, Double bonds mostly in the cis configuration, which create the kinks in the chain. If say for example, this is the straight chain. This for example, this one is the straight chain. This is the straight chain, and here somewhere else in the chain a double bond is introduced. It will bend the chain. The chain will be bent. Melting point in the double point, saturated fatty acids pack in fairly orderly way. Extensive favorable interactions. Unsaturated cis fatty acids pack less orderly due to the kinks. Less extensive favorable interaction. It takes less thermal energy to disrupt disordered packing or unsaturated fatty acids. Disordered packing of unsaturated fatty acids. Unsaturated cis fatty acid have a lower melting point. You can see uh, to be in our native language. अगर हम इसको देखें saturated fatty acids को, तो saturated fatty acids में चूंकि molecules tightly पैक हो जाते हैं, इनके दरमियान में कोई डबल बॉन्ड नहीं है जिसकी वजह से किंक्स क्रिएट हों, तो ये molecules जो हैं एक दूसरे के करीब रहेंगे और एक दूसरे के साथ टाइटली पैक होंगे. इनके दरमियान में हाइड्रोजन बॉन्डिंग इस तरह की जो दूसरी इंटरेक्शन हैं वो डेवलप होंगी और ज़्यादा स्ट्रॉन्ग होंगी. लेकिन अगर हम इसके मुकाबले में अनसैचुरेटेड फैटी एसिड्स को देखें, तो अनसैचुरेटेड फैटी एसिड में जहाँ पे डबल बॉन्ड आ जाता है, वहाँ पे यू कैन सी हियर, वहाँ पे बेंड्स क्रिएट हो जाते हैं. इन बेंड्स के क्रिएट होने की वजह से मॉलिक्यूल्स जो हैं वो थोड़ा सा एक दूसरे से डिस्टेंस पे चले जाते हैं. तो तो जो यू कह लें कि इफेक्ट है डबल बॉन्ड का, लाइक इन दी अनसैचुरेटेड, लाइक इन दी सैचुरेटेड फैटी एसिड्स, वो अनसैचुरेटेड फैटी एसिड्स में नहीं रहता। बॉन्ड्स के दूर होने की वजह से इनकी टाइट पैकिंग नहीं रहती, इनका पैकिंग का ऑर्डर डिस्टर्ब हो जाता है। इस डिस्टर्बेंस इन ऑर्डर की वजह से, इस ऑर्डर डिस्टर्बेंस की वजह से, ये मालिक्यूल एक से दूर होने की वजह से इनका मेल्टिंग पॉइंट लोअर हो जाता है। तो इस वजह से मोस्टली इस तरह के जो मॉलिक्यूल्स हैं वो लिक्विड फॉर्म में होंगे, जबकि सैचुरेटेड फैटी एसिड बिकॉज टाइटली पैक होते हैं, तो मोस्टली ये मॉलिक्यूल सेमी सॉलिड फॉर्म में होते हैं। Trans fatty acids form by partial dehydrogenation of unsaturated fatty acid. Um, uh, this this is to increase the shelf life or stability at high temperature of oil used in cooking. जब कुकिंग ऑयल्स को की पार्शियल डिहाइड्रोजनेशन की जाती है ताकि उनकी शेल्फ लाइफ बढ़ाई जाए, तो मोस्टली हमारा काम हम करना तो ये चाह रहे होते हैं कि उनके दरमियान से जो अनसेचुरेशन है उसको ख़त्म करना चाह रहे होते हैं। लेकिन समटाइम्स ऐसा होता है कि अब बजाय इसके कि अनसैचुरेशन ख़त्म हो जाए, जो बॉन्ड्स की पोजीशन है वो मालिक्यूल्स की पोजीशन वो उनको सिस से ट्रांस में कन्वर्ट कर देती है। तो जो डबल बॉन्ड है बजाय इसके कि वो सिस कॉन्फ़िगरेशन में रहे, वो ट्रांस कॉन्फ़िगरेशन में कन्वर्ट हो जाता है। और ट्रांस डबल बॉन्ड अलाउज अ गिवन फैटी एसिड टू अडॉप्ट एन एक्सटेंडेड कन्फर्मेशन। Trans fatty acid can pack more regularly and show higher melting points than cis forms. तो अब ट्रांस फैटी एसिड्स जो हैं वो एक दूसरे के साथ ज़्यादा टाइटली पैक हो जाते हैं, और इस टाइटली पैक होने की वजह से दे आर देयर मेल्टिंग पॉइंट इज इंक्रीज्ड. Consuming trans fats increase the risk of cardiovascular diseases. Avoid deep frying partially hydrogenated vegetable oils. Current trend reduce trans fats in the food. Um, w- in according to the rules, uh, the fast food shops are the fast food shops must not use the trans fats because these are dangerous to the health. They may create, they may become the cause of many severe diseases like the cardiovascular diseases. So the so these are banned on on such kind of shops, on like the KFC, like the fast food in your city. So the trans fats must not be used. Even in the home, these trans fats must not be used because these are the reason for the cardiovascular diseases. Um, this is the chart showing the trans fatty acids in some typical fast foods and the snacks. French fries contain in typical serving trans fatty acid content 4.7 to 6.1%. Total fatty acid 28 to 36 percent. And if you look on the pizza, 1.1 percent trans fats are present in typical serving, and chocolate bars 0.2%. So this is the chart uh, showing the trans fatty acids in some typical fast foods and snacks. Our next topic will be the types of the fatty acids. The function, today we discussed about the functions and the nomenclature of the fatty acid. The next our topic will be the triacylglycerol, which is the structural, which is the storage lipid. So I'm hoping you will be enjoying to some extent these video lectures. Although the effectiveness of video lecture is not more, or we can say that these lectures are not more effective as compared to the classrooms. But I am hoping it will be I will be able to fulfill to some extent your requirement. Okay, good luck. We will join in our next session in next week, inshallah.